This week and next we are going to focus all about genetics. This week or this unit, we're going to focus a little bit more on DNA itself. So hopefully some of these will be a little bit of review from past courses that you've had, and then we'll get a little bit more information or new information as well. So starting off, just what is DNA? It stands or is short for deoxyribonucleic acid. That's where it gets its D, N, and A. And it is made up of repeating nucleotides. Nucleotides are the building blocks that, when we put them together, construct the actual DNA molecule itself. Now, the nucleotides, the building blocks that we use, have three parts to it. They have a sugar, and the sugar is called deoxyribose, which is where it gets part of its name. It has a phosphate group, and then it has a nitrogenous base. Now, two of the nucleotides, there's 4 different nucleotides. So there's only 4 different nucleotides, and then based on just their order is where we get our DNA sequence from. So out of the 4 different nucleotides, out of the 4 different building blocks, 2 of them are called purines and 2 of them are called pyrimidines. And it has to do a little bit with that nitrogen base that all these 4 different nucleotides have. Again, a nucleotide has a sugar, a phosphate, and a nitrogen base. And that nitrogen base is either going to be adenine, guanine, thymine, or cytosine. Adenines are a little bit smaller, pyrimidines are a little bit bigger. Another way how I like to remember them on which ones are which, pyrimidine has the letter Y in it, as does thymine and cytosine. Now, DNA uses complementary base pairing to store all of its genetic information, meaning you're always going to have one purine pairing with one pyrimidine, and it's always going to be the same nucleotides that pair up. Adenine will always pair with thymine. Guanine will always pair with cytosine. How to remember them? A and T together spell the word at. The letters G and C almost do look a little bit alike. I've also heard other ways to remember that C could stand for car, G stand for garage. Cars go in a garage, so they go together. Either way, adenines will always pair with thymines, guanines will always pair with cytosine. That's what it talks about complementary base pairing. Now, each human chromosome contains, on average, about 140 million of these base pairs. And so the number of possible nucleotide sequences, the fact that you have 140 million base pairs, and they could be one of each of these 4 choices, is 4 to the 140 millionth power. So quite a few different possibilities on the different DNA structures and sequences. Now, just showing a little bit zoomed in on these different parts of the DNA. As I said, all the nucleotides, those building blocks, are made up of a phosphate group, a sugar group, the deoxyribose, and a nitrogen base. This is showing one nucleotide here that has guanine as the nitrogen base. This on the other side is showing another nucleotide, but it has cytosine, and G's and C's always pair up. Also, another little bit just on its structure alone based on this picture. So we've got our complementary base pairs. They do hang out in the middle. So the sugar phosphates always hang out on the outsides. They call it the backbone. And then the nucleotides are attached in the middle, and they're attached just by a hydrogen bond. I mentioned that because hydrogen bonds are a quite weak bond, which works out really well when we need to separate the DNA apart. Also, as we can see kind of the basic structure, you may also notice that these look like they're almost upside down. Well, they are a little bit, and that's because DNA runs what's known as anti-parallel. So one strand of the DNA runs, we call it 5' and 3' for the ends. One end is the 5, the other end is this 3, and because it's opposite on the other side, that means on the side that this has the 3, this one has the 5, and vice versa. They go opposite of each other. And so we are double-stranded, we are anti-parallel, those 2 strands go in opposite directions of each other. Sugar phosphates are on the outside, the nitrogen bases on their inside and are pairing up by a really weak bond. And when this pairing up happens, although they are two strands that are connected together, the pairing actually causes a twist to happen in the DNA into a helical shape. Now, we didn't magically know that A's paired up with T's and G's paired up with C's. Chargaff was the man that discovered this. He started to look at the number of percentages of all the bases that were in the DNA. How many of them were A's, how many were T's, how many were G's, how many were C's. And you notice that comparison, that the percentage of A's is almost identical to the percentage of T's, and the percentage of G's is almost identical to the percentage of C's, which makes sense because they pair up with each other. Now, we still also didn't know the shape of DNA right away, so we still had to do a little bit of research, and Rosalind Franklin was the scientist that discovered that DNA has that helical shape to it, that it's not just one long strand, that it can actually twist and turn. So she discovered that it's a DNA shape. And I should point, I'm like, although DNA will look like a strand, in the virtual lab in this unit, you get to actually kind of separate out some DNA into strands. So you can actually see its strands, but it is actually helical shaped. Watson and Crick were the ones that in the 1953, they finally kind of put everything together. They took the information from Rosalind Franklin and from Chargaff, and they eventually were able to build how this looked. They figured out that it has a helical shape. They figured out that it was anti-parallel. They figured out that the C's paired with T's and A's paired with G's, and they put this all together to actually build what the structure looks like. And they received a Nobel Prize a little less than 10 years later for developing and figuring out this full structure. Here's an actual picture of them building and developing this model to demonstrate how these all paired up and built together. Now, when we need to make more DNA, because every time a cell replicates itself through mitosis, it has to double the amount of DNA. That's part of what happens in that interphase. We have to duplicate the DNA before the cells can divide. So we're going to take a look at that process of DNA replication. So we are copying some of the DNA. So all the information is going to be the exact same. We're just making a copy of it. However, how DNA replicates is they had to figure that out as well. There is a whole process, and they didn't quite know exactly how it replicated right away. With enough research, they figured out that it replicates in what's known as a semi-conservative replication. So you do have a double-stranded molecule. Well, what the semi-conservative means is that each of those original strands becomes a template for each new daughter strand. So shown on this picture, I like the just blues and reds. My original strand is blue. Well, we can separate out those two blue strands that make up our original model, and then each strand becomes a template to make a copy for the other strand. And so we have our one original strand, and then at the end of DNA replication, we have two strands, but each of those DNA strands still has part of or half of that original strand. So that's what's considered the semi-conservative replication. Now, what's required to make DNA? Well, first we have to unwind those two strands of DNA. Luckily, as I said before, they're held together by pretty weak hydrogen bonds, so it's not too hard to unwind them. We also then have to have complementary base pairing. If there was a T on the original strand, you would have to bring in an A, because A's and T's pair together. If there was a G on an original strand, you would have to bring in a C, because C's and G's pair together. And then we have to join those two nucleotides back together and join them up with hydrogen bonds so that we ultimately, in the end, have two DNA molecules, and each one has an old original strand, and each one has a new strand. But what about prokaryotic cells? How do they do this? Do they do it any different? Not much. The big difference is just what their DNA looks like. The DNA in a bacteria, in a prokaryotic organism, is circular. It's not a long helical strand. It's circular as shown. So when they make new DNA, they still have to unwind it. They're going to start in one spot. It's called the origin of replication. And they pull the two pieces apart as they go along. They don't pull the whole thing apart. And the new green nucleotides are what are getting laid down. So as it starts to open it up and it starts to unwind, and it unwinds going in both direction, it lays down nucleotides going in both direction. And it will continue to unwind going around the entire thing until ultimately you'll have the same type of idea, that same semi-conservative model. You'll have two circular strands of DNA. Each of them have an original template strand and each have a brand new strand. Now, replication in a prokaryotic cell can take about 40 minutes to happen, but the cell divides every 20 minutes. So that doesn't work. You can't divide until your DNA is present. So to solve that problem, they can actually start a round of replication before they're done even dividing, or before they even go into that mitosis type of phase. So a new round of replication can begin before the previous round is even done. So while this strand is being made, right here or right here, you could have replication already beginning. It allows the bacteria to replicate very, very quickly. They don't have to wait for this whole replication to take place. They can just keep replicating even while they're replicating. Now, replication of DNA in eukaryotes, because we have that long strand, again, we just kind of have those three basic steps of unwinding, adding the new nucleotides, and putting them back together. That's the basics of DNA replication. It gets a little more complicated than that. So what can speed it up, even in eukaryotic cells, we don't have to only start replication in one spot. We can actually do replication in multiple places. That helps speed up how fast we can make DNA. But we are still going to have to unwind it, unzip those two strands, break those hydrogen bonds. We're still going to have an original template. We're still going to have the new DNA, those new nucleotides coming in. They're still going to be matching up A's with T's and C's with G's. The DNA that helps bring in those A's, T's, C's, and G's and helps make the new DNA is an enzyme called DNA polymerase. So it's really helping bring in all the correct nucleotides. It helps propagate that elongation and continuation of making the new DNA strand. So extremely important enzyme that's needed. Now, as it does replicate in multiple places, you do get what are known as replication bubbles. It kind of does look like little bubbles on here. Ultimately, as the bubbles start to meet together, they'll eventually get to their full brand new full-length DNA strand. And again, that conservative method or conservative model that are at the end of DNA replication, we will have two strands. Each of them have an original strand and each of them have a brand new strand. Now, just a quick little review. If your original strand, so we'll go with the dark teal on here, contained the bases AAT, CGC, what would the complementary strand be? What would be the nucleotides? What would be those bases that would get brought in? So if you had three letters of AAT, the three bases that would come in, if you had an A, we would have to bring in a T. If we have another A, we'd have to bring in another T. If we have a T, we'd have to bring in an A. If we have a C, we'd bring in a G with a C, C with a G, and so on. So your complementary strand would have the TTA to go with opposite of AAT, and to go opposite of CGC, you'd have GCG. Again, whatever the original strand is, it would bring in what was complementary or opposite of it. This is just kind of showing the difference on what the prokaryotic replication looks like compared to the eukaryotic replication. Again, prokaryotic replication, it has that circular DNA. Ultimately, you still make two new strands, both semi-conservative, but they're circular, whereas where eukaryotic cells, they have a linear DNA, but we can start that replication in multiple places. Now, it's not always quite that easy. There's always, a quick twist of okay, it's not just bringing in the bases, it's not just unzipping, it's not just putting it back together when you're all done. DNA polymerase, when it brings in those correct bases of A's to match with T's or G's to match with C's, it can only do it on the 3' side. So when you have that anti-parallel DNA, so each of these strands is running in opposite directions. So on one strand it'll be the 3, which means the other new strand that we're going to make is going to start with the 5. When we rip this apart, each of these strands is going in opposite directions. And so the first strand, we're going to call it the leading strand. The end of it is the 3 side, which means our new strand that we are going to be making. So we're unzipping going to the left on here. And as we are unzipping, we have the enzyme bringing in the correct bases. A's to pair up with T's, G's to pair up with C's, and so on. And it can do it because it is adding it to the 3' side of this new strand. Because it started off 5 over here, which means the 3 is over here. And that's fine, because as we are unzipping going to the left, this is just chugging along, adding a nucleotide to nucleotide to nucleotide on that 3' side of this new strand. But the lagging strand has an issue. Its strand, it ends in 5 over here, and the new strand has the 3 over here. And the polymerase can only add onto a 3' side. It can't add on to the 5' side of this new strand. So to solve that problem, it makes little mini fragments going backwards. So it's going to start off always with a little primer, and then it's actually going to add bases going to the right. So we're separating DNA going to the left, but we're going to make DNA going to the right. So it will make a little fragment here. It would make another little fragment. So as we are constantly separating the 2 strands of DNA, it keeps laying down a little primer and making DNA reverse. Ultimately, it will get rid of these primers and fill in the correct bases, but it has to keep going backwards. So they call it the lagging strand because it does take a little bit longer to go backwards, but it generally works pretty well. So if I can get this video to play, we're going to play a little bit of this video that you can actually see the difference between the leading strand and lagging strand. Video-wise, it does talk about some different enzymes that you're not responsible for. I'm only looking that you recognize DNA polymerase.
When DNA replicates, its strands are separated by the enzyme helicase. Single-stranded DNA binding proteins keep the strands from reannealing. One DNA strand encodes the leading strand, which forms from its 5' to its 3' end, using DNA polymerase 3. No problem here, but the lagging strand presents problems. It has to form from 5' to 3' too. It forms in pieces called Okazaki fragments. First, an RNA primase lays down an RNA primer. Then, DNA polymerase 3 lays down new DNA. The process repeats again and again. DNA polymerase 1 replaces the RNA primers with DNA. Finally, DNA ligase links the Okazaki fragment.
I'm just going to make sure it pauses right there. Again, the leading strand, never an issue. It can keep laying down new bases in the same direction that it unzips. But it's that lagging strand, as it goes backwards, it actually has to make the DNA backwards in those little Okazaki fragments. And eventually it does put them all together, so it's not fragmented, but it's a little trickier. Now, DNA polymerase that brings in these new bases is pretty accurate, yet it makes mistakes about once every 100,000 base pairs. So if it sees a T and it's supposed to bring in an A, maybe it accidentally brings in a G or a C instead. Luckily, our cells do have checkpoints that can catch when that happens. It can catch when it has those mistakes, because those mistakes themselves are then considered a mutation. And depending on which base, how many mutations occurred, and which cells that it happens in, it could be a fatal mutation. So luckily, our cells are capable of going back in and correcting that. Now, when we make our DNA, we saw this a little bit when we kind of went through the mitosis. Our DNA is that double helical shape, but when it goes into mitosis to start dividing, that DNA condenses into visible chromosomes, and you can start to see those chromosomes in that prophase. Well, how does it take that long, skinny DNA strand, because it is skinny, and get it condensed enough that it then is visible, that we can see easily underneath a microscope as those characteristic X chromosomes? So I've got a little video that's going to show that as well. Now, before I start it, this pinkish color, that's the DNA. That's that helical shape. And there are proteins called histones that help this DNA wind up. And a lot of it is winding, it is winding, it is winding until it becomes thick enough that we can see it.
In this animation, we'll see the remarkable way our DNA is tightly packed up to fit into the nucleus of every cell. The process starts with assembly of a nucleosome, which is formed when 8 separate histone protein subunits attach to the DNA molecule. The combined tight loop of DNA and protein is the nucleosome. Multiple nucleosomes are coiled together, and these then stack on top of each other. The end result is a fiber of packed nucleosomes known as chromatin. This fiber, which at this point is condensed to a thickness of 30 nanometers, is then looped and further packaged using other proteins which are not shown here. This remarkable multiple folding allows 6 feet of DNA to fit into the nucleus of each cell in our body, an object so small that 10,000 nuclei could fit on the tip of a needle. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes. It is important to realize that chromosomes are not always present. They form only when cells are dividing. At other times, as we can see here at the end of cell division, our DNA becomes less highly organized.
Winding up, till it becomes that visible chromosome that we can easily see it separate and go through those stages of mitosis. So cool. Now, we know the basic structure of DNA. We know how to replicate and make more of it. But what is in the DNA, and how do we get that information that's in the DNA translated into something useful? So that's when we get into what's known as the central dogma, the genetic code of life. The fact that we have all of these bases that are paired up in 4 times the 140 millionth power of options. That information that's in that DNA codes for something. And we have genes that are found on the DNA. So DNA is grouped in genes. A gene is just a small snippet of DNA. So 140 million base pairs, we can talk about, well, how about just this 1,000 base pairs? This one little tiny 1,000 base pair section. And that little tiny 1,000 base pair section, that is a little chunk of DNA that codes for something. It codes for some important information that we need somewhere in the body. And that little snippet of information needs to be expressed. We need to get the information from it to start making some type of protein. So the idea is that that information is in the DNA, and the information that comes from it is expressed through some type of protein. So the DNA that's in any particular gene, a small little snippet of the DNA, determines the sequence of nucleotides. So those 1,000 nucleotides in the genes is a very specific sequence of nucleotides in that gene. And those nucleotides in that one little 1,000 base pair segment controls the nucleotides that would be in RNA. And we're going to talk a little bit about RNA coming up. And RNA is what can be used to then give us the protein that's needed. So we're going to go from the information that's in DNA and translate it out into RNA, and then we're going to translate it further into the correct protein that our bodies need. So what that looks like is a DNA is a double strand, and that's fine, but we need to be able to take one small segment of the DNA. And on here we've got 9 bases, so really small fictional segment. And we're going to copy one of those pieces of information. So we're going to copy this strand. So if the original DNA strand was a T, we would bring in an A. If it had a C, we'd bring in a G. If it had a G, we'd bring in a C, and so on. So we are just making a copy of one strand of DNA. Now, this making a copy of the DNA happens inside the nucleus. The nucleus contains the DNA, and that DNA is extremely important, that it needs to stay protected inside of the nucleus. So if we're going to make a copy of the DNA, we have to go where the DNA is found, and that's inside the nucleus. So now we have this copy of these 9 bases. We now have messenger RNA. That's what the little M stands for. And this messenger RNA can find itself a ribosome, and ribosomes are known for making proteins. And that ribosome can read that messenger RNA, and it can translate it into the correct amino acids that make a protein. And we're going to take a little bit more in-depth look at what's happening during this transcription and what's happening during this translation. But first we're going to back up just a little bit, just to talk a little bit about RNA and compare it to DNA. So RNA is a polymer. It's made up of repeating units of RNA nucleotides. Just like DNA was made up of DNA nucleotides, the A's, the T's, the C's, and the G's, RNA is made up of RNA nucleotides. They're very similar except one. It still has cytosine, it still has guanine, it still has adenine. The only thing it's missing thymine. Instead, it has a very close cousin to thymine called uracil. And structurally, uracil and thymine do look similar enough, so A is going to pair up with U instead of a T, because we don't have T's around. It still is also going to have a sugar, except it's ribose instead of deoxyribose, and where it gets its name of ribonucleic acid versus deoxyribonucleic acid. So the biggest difference, though, is that it has a U instead of a T, and RNA is always single-stranded. Now, there are also 3 types of RNA, and we're going to focus on 2 of them in this rest of this PowerPoint. Messenger RNA, because it's carrying the message from the nucleus out into the cytoplasm to go find itself a ribosome. We talk briefly about the ribosomal RNA. So the ribosome's job is to make protein, and that ribosome organelle itself does contain ribosomal RNA. But transfer RNA plays a big role, because transfer RNA, something has to bring those amino acids in, and that's what tRNA, that transfer RNA, does. So we're going to focus mostly on messenger RNA and transfer RNA. This is showing the structure of RNA. It is, again, only single-stranded. It still has a phosphate, it still has a sugar, and it still has a nitrogen base. The only big difference is that its nitrogen bases are GU, A, and C. So some similarities, differences to DNA. They both have that same type of backbone of a phosphate sugar backbone. A difference, though, it's a different sugar. They do both have the nitrogen bases. However, one difference is that it has uracil instead of thymine. Also, DNA is only found in the nucleus, where RNA can leave the nucleus and go out inside the cytoplasm, which is what we want it to do. So, making protein. So protein synthesis in a nutshell. DNA codes it codes for making a protein. It has the information to make any protein our body needs, but DNA can't leave the nucleus. So we have to make that messenger RNA. It will transcribe the message. It will make a copy of a message, and it will leave the nucleus and find a ribosome. And then at that ribosome, transfer RNA is going to translate that new message into amino acids. When we join all the amino acids together, we make a protein. It's like, so simple. But we'll get into a little bit in depth. So during transcription, we first have our original DNA. That was double-stranded DNA. We have to unzip it so that we make a copy of just one of the strands. So on here, we're only going to make a copy of the bottom strand. So if the bottom strand had a T, we bring in A. If it had an A, we bring in a U, because we don't have T's in RNA. If it had a C, we bring in a G we bring in a C, and so on. And so we're bringing in those correct nucleotides, C's pairing with G's, A pairing with U's. When the entire gene, when we have this entire messenger RNA transcribed all the way to the last letter, we have what's known as a pre-messenger RNA transcript. Pre is because we still have to modify it a little bit before it actually goes out and gets translated into amino acids. And the base sequence that makes up this messenger RNA is complementary to the DNA. Just saying that they've got the T's matching with A's, A's with U's, C's and G's. It is a copy of the original DNA. This again is showing we have our original strand where we unwound it, and if the original DNA strand had a T, we brought in an A. If it had a G, we brought in a C. If it had a C, we brought in a G. If it had an A, we brought in a U, not a T, because this is going to be our RNA strand. Now, when we read DNA, we generally read DNA 3 letters at a time, and every 3 letters is called a triplet. When we read RNA, we also read them 3 letters at a time. We call them codons. Same type of meaning, it's just every 3 letters generally codes for something. Now, I'm going to watch just the first part of this video that's going to show that kind of ripping apart of the DNA and starting to make that original messenger RNA. So this is going to be happening, it's in real time. So it goes quickly, but it's kind of cool. And it's going to show inside the nucleus us making that new messenger RNA strand. This is your original DNA strand there.
The innermost workings of how a simple code is turned into flesh and blood. This is what Francis Crick called the central dogma of modern biology: how DNA makes protein. It starts with a bundle of factors assembling at the start of a gene. It's these that trigger the first phase of the process, reading off the information that will be needed to make the protein. The gene is the length of DNA stretching away to the left. Everything's ready to roll. 3, 2, 1. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the 2 strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to make an exact copy of the A's, C's, G's, and T's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related nucleic acid known as U. You are watching this process, called transcription, in real time. It's happening right now in almost
Alright, I'm going to
every
pause it
cell
right there
in
and
your
we'll
body.
actually finish the video shortly. So we just got done making that new messenger RNA strand, that
When
big
the
yellow
RNA
strand.
copy is complete,
Make a copy of it. All
it snakes
happens
away
in
from the
the
nucleus.
nucleus. That's the process of transcription, of making a copy. So if your original DNA sequence consisted of TAC, GCA, CCA, what would the RNA sequence be? Again, if it has a T, we would bring in an A. If it has an A, we'd bring in a U, because RNA doesn't have T's, it has U's, C with G, and so on. So your 3 codons, I grouped them purposely in groups of 3, you have AUG, CGU, GGU. However, this new strand, this pre-messenger RNA, does have to get modified a little bit before leaving the nucleus. It has to get a couple things changed before it's really ready to be read. First, on the 5' end, we add what's known as a cap. It's got a guanine that we add on. It's almost like a big red flag for the ribosome to know where to start and attach. On the other end, we add a whole bunch of A's. We add a whole bunch of adenines to the 3' side. And that poly A tail, that big long group of A's at the end, helps that messenger RNA leave the nucleus, and it helps prevent it from being broken down by enzymes. So we modify it a little bit by, adding a little bit to each end. Also, that messenger RNA has what are known as exons and introns. Any little section on that message that's known as an exon is information that's going to be expressed. We're going to read it, we're going to make the protein from it. However, there are some introns in between, and the intron just literally means they are in between the exons. And it allows the cell to pick and choose which exons are going to go into the correct messenger RNA and actually be read. So we do have a structure known as a spliceosome that can remove the introns because they don't actually express or do anything, and it leaves the exons there to be expressed. But if needed, it can move a few things around as well. It can actually move exons around and switch around a little bit of the information. But once we've removed all the introns and we've moved around the exons where we need them to go, we've added that 5' cap, we added the poly A tail, now it's finally ready to be able to leave the nucleus and go do its jobs. Now, I usually get the question is, why do we care about these introns if they're just getting spliced out and removed? Well, the exons, they are expressing information, and depending on kind of where they are and what information is on them, the exons might combine into different combinations and give us different information. The introns found in between might regulate which genes we want expressed and read, and it looks like it may, based on information that we know, encourage some crossing over during meiosis. Again, it gives us another place where we can have a little bit of genetic variance. So just my little recall on central dogma so far. So we've got our double-stranded DNA that stays inside the nucleus, but through transcription, we're going to make a copy of just one of those strands and make it into that messenger RNA. We'll modify it just a tad, it will leave the nucleus, and that's where we're going to go into translation. And the ribosome's job is to make proteins, and so that's what's going to end up reading this new message. So translation, we have a messenger RNA. It's going to associate itself with the ribosome or RNA of a ribosome. It just means the ribosome is now going to be able to read this new message. The ribosome reads the information on that transcript. It directs transfer RNA to bring in the specific amino acids that it needs to bring in, and it does that. It reads the messenger RNA 3 letters at a time. So AUG tells transfer RNA to bring in methionine. CGG tells transfer RNA to bring in arginine. So it directs exactly which amino acids get brought in. So it's determined by the code, those 3 letters at a time. So the sequence of codons, every 3 letters, so I've got these 3 letters methionine, these 3 letters arginine, these next 3 letters tyrosine, next 3 letters leucine, and so on and so on and so on for thousands of bases and thousands of amino acids long. We eventually are going to have this long chain of amino acids, which we call as a polypeptide. When we take that polypeptide and we shape it correctly, that's when we get a protein. Now that transfer molecule that brings in the correct amino acid. So if it sees AUG, it's going to bring in methionine, but there's a reason why it knows to bring methionine. Is because on the transfer RNA itself, it can recognize any codon that's on the RNA because it has the opposite letters. So if on your RNA you have CUU, on the actual transfer RNA itself, this is two different schematics of transfer RNA, it has a 3-sequence letter that recognizes the codon, those 3 letters, because it has the opposite letters, so we call it an anticodon. So an anticodon for methionine would be UAC. So the transfer RNA would actually have UAC on it. It would recognize AUG and it would bring in methionine. So it's not just a random choosing that transfer RNA knows which amino acid to get brought in. Now, there are 20 different amino acids that can make up any particular protein. So there's 20 different amino acids that can get brought in. But theoretically, because we have 4 different bases and they are read 3 letters at a time, there are 64 different ways you can mix C, G's, A's, and U around. And so we do have it sometimes like CUU and CCU could both code for the exact same amino acid because there's 64 combinations but only 20 different amino acids. Now it's the ribosome that is doing all of this reading and translating. Its job is protein synthesis, and that's what's happening in translation. And so it is made up of ribosomal RNA that eventually it was made at the DNA at some point. And it's inside this ribosome, this big yellow blob, that we pull that messenger RNA through like a tape. We just pull it through and it goes through 3 sites. There's an A site at the very beginning. I'm kind of reading it backwards because it's coming through this way. It almost spells ape backwards. I always think of A for protein. It's coming in at the A site. That's where the amino acid is getting read. The P site is for the peptide. That's the bond that's going to put amino acids together. And the E is for the exit because the messenger RNA is going to leave. So this again is showing that same picture, but now we could actually see it in different stages. So the messenger RNA is getting pulled through and it's reading the amino acid here. The transfer RNA is actually assembling it and adding that peptide bond, and we can actually see this new chain of amino acids coming out of the ribosome. Now, as shown on here, you can have one messenger RNA segment that's going to code for some particular protein. You don't have only one ribosome reading it. You can have multiple ribosomes reading it so that you can crank out a ton of proteins very, very quickly. Here's an actual protein that's showing that. This is a zoomed in picture showing those 3 sites, your A site, your P site, and your E site. That P site is where that transfer RNA is actually bringing in and attaching the different amino acids, and the E is because it's leaving the ribosome at that point. So again, just another schematic showing you've got the A site going in, we've got at the P site, you've got the different amino acids that are getting bound together with a peptide bond, and you can see this nice long chain of amino acids starting to grow out the top of the ribosome. Whenever it gets to the end, there is a specific codon, 3 letters, that there is no amino acid that gets brought in, and so it's considered a stop codon. And so once the ribosome reaches one of those, it's done reading. The ribosome actually breaks apart, the RNA goes away, but what we end up with is we have our polypeptide, we have our protein chain. Now we're going to continue that same video that we started, but I'm going to get to right where we left off. So we saw that new messenger RNA getting made. Now we're going to see it getting translated. So we're going to see the ribosome chomping down I think it looks like it's jumping down.
The
And
innermost
that messenger RNA, and you're going to be able to see the transfer RNA. There's the ribosome, big blue blob.
breaks away
You're
from
going to
the
see the transfer
nucleus
RNA
and
getting
into the
brought
outer
in
part of
and
the
bringing
cell.
all the little red amino acids and assembling them together.
Then, in a dazzling display of choreography, all the components of another molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. They all carry a specific 3-letter code that will be read by the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecule. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed
I
in
love that because
on
you
the
can truly
RNA.
see how fast that can happen. And you have to
In this
remember there are
case,
multiple
the end product is
ribosomes all doing
hemoglobin.
that exact same
The
pace
cells in our bone
on
marrow
that same
churn out
messenger
100
RNA.
trillion molecules
So,
of it per
just my
second.
last kind of little bits on how does the ribosome know what amino acid to bring in. Again, there are 4 different bases that are grouped together into groups of 3 letters. That's your codon. And each codon codes for a particular amino acid, and there's only 20 amino acids. So 4 different bases, 3 bases in a codon, 20 amino acids equals 64 different combinations, but there's only 20 amino acids. So just showing an example, AUU, AUC, AUA, all code for the exact same amino acid. You can see some redundancy that some similar codons are going to code for the exact same amino acids. Again, 64 combinations, only 20 different amino acids. We're going to have some redundancy. But I also wanted to point out that AUG, this particular 3-letter sequence, is what's known as the start codon. This is where ribosome likes to start reading that message. There are, however, 3 stop codons. So these are 3 different codons that there is no amino acid that gets brought in, and so when the ribosome encounters one of these stop codons, it stops replicating right then and there, and it's done making the polypeptide chain. It's done making the protein. It's as long as it needs to be. So a quick little review. If your DNA sequence consisted of the TAC, GCA, CCA, what would the RNA sequence look like? So you're going to see some questions like this on your next quiz. So if the DNA sequence was TAC, the RNA sequence would be AU, because it's RNA, AUG, and then it would be CGU, again, that's what the RNA sequence is, and then GGU. The polypeptide chain, you would actually have to go back and find AUG on here, which codes for the amino acid methionine. CGU, this is the first letter is the C. What was the second letter? I apologize. CGU. The first letter is C, the second letter is G, and the third letter is U. So CGU codes for arginine, and then GGU, first letter's G, second letter's G, third letter's U is glycine. It tells us which amino acids get brought in. So we have our methionine, our arginine, and our glycine. Makes our 3 little polypeptide chain. So this again is just our last slide to kind of end up with a little bit of summary of that protein synthesis. that you're going to have transcription happening, making that initial messenger RNA inside the nucleus. We get rid of the introns, don't need them, and then that finished off messenger RNA will leave the nucleus, find itself a ribosome. The ribosome attaches, it pulls that message through, it reads it 3 letters at a time, so every 3 letters is a codon. The transfer RNA brings in the correct amino acid, and it starts to assemble those different amino acids, adding peptide bonds to assemble them. Ultimately, when it's done, we have this long chain of peptides, and we can fold it up into the correct protein.